Editing our DNA to remove sickness or change traits sounds like technology from a science fiction movie, but it's real and it's becoming increasingly more common. It's called CRISPR, and it's changing what we know about our bodies and genetics forever.
While gene editing itself has been around since the 1980s, CRISPR-Cas9, developed in the early 2010s, is far more precise, efficient, and affordable than anything that came before it. It has quickly become the go-to method for altering DNA in labs around the world. While seemingly a fantastical technology, it actually traces its roots back to biology, specifically to bacteria and their defenses against viruses. When infected by a virus, bacteria will snip off a piece of the virus's DNA and bind it to its own genome. This puts the virus's DNA in a sort of memory bank. The memory of the virus is then transcribed into what is known as CRISPR RNA or cRNA. This CRISPR RNA is then attached to a protein called Cas9. Cas9 operates as the scissor enzyme used to cut DNA segments. If infected with the same type of virus, the bacteria's CRISPR-Cas9 can recognize the virus, locate it, and then cut its DNA, rendering it ineffective. Technology has allowed us to hijack this process and turn it into a controllable tool with nearly endless possibilities. By modifying the cRNA into what is known as a guide RNA, scientists are able to attach any DNA sequence to the Cas9 protein. This means, if programmed to do so, the CRISPR-Cas9 can seek out and cut the DNA of any desired trait. This would allow the gene to be edited, replaced, or turned off entirely. The medical potential is enormous. CRISPR is already being tested as a treatment for genetic disorders like sickle cell anemia, muscular dystrophy, and certain inherited forms of blindness. In some cases, results have been astonishing. Patients with once incurable conditions are now showing signs of lasting improvement. In cancer research, CRISPR is helping doctors reprogram immune cells to better recognize and attack tumors. Scientists are also investigating how CRISPR might be used to slow or even prevent the progression of conditions like Alzheimer's. But the technology doesn't stop at curing diseases. CRISPR has also been used in agriculture to create crops that are more resistant to droughts, pests, and disease. It's even been used to revive genes from extinct species, like the woolly mammoth, in what some researchers call de-extinction. Still, perhaps the most controversial use of CRISPR is in editing human embryos. The idea of designer babies, children whose traits like intelligence, appearance, or athletic ability can be selected and enhanced, has alarmed ethicists and lawmakers alike. There are fears it could lead to a genetic divide where only the wealthy can afford to have the perfect child. Regulations around CRISPR vary widely. Some countries have banned gene editing in embryos altogether, while others allow it for research but prohibit implementation. The debate is ongoing and growing louder as the science moves faster than policy can keep up with. What's clear is that CRISPR has already begun reshaping our future. It holds the potential to eliminate the suffering for millions, but it also challenges us to ask, just because we can, should we? On a more personal note, and having done a lot of research on this topic, I tend to be a supporter of CRISPR technology for all it's worth, both good and bad. I typically tend to be of the opinion that any advancement in science is an advancement worth exploring, that the resulting ethical conundrums are necessary evils to further the advancement of humanity. Intelligence, I think, is the most beautiful aspect of humankind, and for it to be constricted, for innovation and progress to be halted, because of purposes derived from societal issues is, in my opinion, the greatest tragedy possible. However, while I consider them necessary evils, I still consider them evils. Legislation should be used to maintain the good of progress while minimizing the bad. Stay with us. More news after the break.